This video is for The Living Earth, Unit 4.3, Transcription and Translation. Today's essential question is, what are the functions of mRNA, tRNA, and rRNA, and how do they work together to make proteins? This question guides everything we'll explore. To answer that, we'll need to understand what proteins are and why they matter. Proteins are the molecules that do most of the work in your body. They help build muscles, digest food, fight infections, and even help you think by controlling brain signals. Now, where do these proteins come from? They are not made randomly. They are built from instructions that are coded in your DNA. But your DNA never leaves the nucleus. That's where RNA comes in. RNA acts as a middleman between DNA and the machinery that builds proteins. There are three main types of RNA, as we've discussed previously, and each one has a special job. mRNA is like a messenger that carries the code from DNA. tRNA is like a delivery truck that brings the correct building blocks. And rRNA is like the machine that puts everything together. Today we'll learn how they interact in a step-by-step process to make proteins, starting in the nucleus and ending at a cellular factory called the ribosome. Remember, RNA stands for ribonucleic acid. It's a type of molecule found in all living cells, and it's closely related to DNA, which is your body's master instruction manual. But unlike DNA, which stays safely inside the nucleus, RNA is more like a worker that goes out into the cell and does jobs. Think of DNA as a locked filing cabinet that stores all the information about how to build and run a cell. RNA is like a copy of a specific page from that cabinet. It can go out, deliver instructions, and help build what's needed, especially proteins. Without RNA, the information in DNA would never leave the nucleus, and proteins, the tools and machines of life, would never get made. So let's get into it. Just like a construction site has different workers, like architects, delivery drivers, and builders, cells use different types of RNA to get the job done. So let's take a look at each type. mRNA, which stands for messenger RNA, carries a message. Specifically, it carries a copy of the genetic code from the DNA, which is in the nucleus, out to the ribosome, where proteins are actually made. There's tRNA, or transfer RNA, which brings the correct amino acids, remember these are the building blocks of proteins, to the ribosome. There's rRNA, which stands for ribosomal RNA, which is a part of the ribosome itself, which acts like a protein factory. rRNA helps assemble the amino acids into a chain. Together, these three types of RNA read, interpret, and build proteins, just like a coordinated team. DNA and RNA are both made of the same basic building blocks, which are called nucleotides. But they have important differences that help them to do different jobs. Recall that DNA is a double-stranded helix. This shape makes it very stable, which is perfect for long-term storage of genetic information. DNA has a sugar called deoxyribose and uses the base called thymine, which is represented by a capital T. RNA, on the other hand, is a single strand. It uses a slightly different sugar, which is called ribose, which has one more oxygen atom, making RNA less stable and more reactive. This is actually a useful thing because RNA's job is temporary. RNA also uses uracil, which is represented by a capital U, instead of thymine. So when you see a U in a genetic sequence, you know you're looking at RNA. Messenger RNA, or mRNA, is made in the nucleus through a process called transcription. An enzyme called RNA polymerase reads the DNA and builds a complementary strand of mRNA. But before the mRNA leaves the nucleus, it has to go through processing. It's like editing a rough draft. This includes adding a protective cap and tail, and removing any non-coding segments, which we call introns. The parts that actually code for proteins, which are called exons, are then spliced together. Then, the mature, complete mRNA leaves the nucleus and heads into the cytoplasm, where it finds a ribosome and begins the next step, translation. Now, let's zoom in on the language of mRNA. mRNA is read in sets of 3 nucleotides, and that set is referred to as a codon. Each codon corresponds to a specific amino acid. For example, the codon AUG tells the ribosome to start building a specific protein and also codes for a specific amino acid. There are 64 possible codons, but only 20 amino acids. So, multiple codons can code for the same amino acid. That's called redundancy, and it helps prevent some types of mutations from causing problems. Some codons tell the ribosome to stop building the protein. These are called stop codons. Transfer RNA, or tRNA, has a special shape that lets it do 2 important things. First, it has an anticodon, a set of 3 bases that pairs with a codon on the mRNA. Second, it carries an amino acid, which is the building blocks of all proteins. When the tRNA matches up with the correct codon on the mRNA, it places an amino acid in line. The ribosome links these amino acids together one at a time, forming a chain that will later become a protein. After delivering that amino acid, the tRNA goes back into the cell to pick up another one. This recycling helps the process happen quickly and efficiently. Ribosomal RNA, or rRNA, is a key part of the ribosome, which is the structure where proteins are made. The ribosome has 2 subunits, and rRNA helps hold these together and make sure everything is lined up properly. But rRNA does more than just hold things in place. It also acts as an enzyme, helping create peptide bonds between amino acids. These bonds are the bonds that link the amino acids into a growing protein chain. Because of its central role, rRNA is one of the oldest and most conserved molecules in biology. Life as we know it wouldn't exist without it. Translation is the process of building a protein from an mRNA strand. It begins when a ribosome binds to the start codon on mRNA, which is usually an AUG. Then, tRNA brings the correct amino acids, matching the codons with their anticodons. As the ribosome moves along the mRNA, it links the amino acids together with peptide bonds. When it reaches a stop codon, the ribosome releases the completed protein and the mRNA. The protein can now go on to perform its job in the cell, like building tissue, carrying signals, or fighting infections. Many ribosomes are attached to a structure called the rough endoplasmic reticulum, sometimes called the rough ER. This organelle looks bumpy under a microscope because it is covered in ribosomes. Proteins made here often need extra steps before they're finished. They are later folded into the right shape, sometimes modified, and then packed into vesicles to be shipped to where they're needed. Some cells, like those that make insulin or antibiotics, they have a lot of rough ER because they need to produce large amounts of proteins quickly. But not all amino acids are the same. Some are hydrophobic, meaning they avoid water, and they cluster together inside the protein. Others are hydrophilic, meaning they're drawn to water and stay on the outside. Some are charged and help proteins interact with other molecules. These traits just listed affect how the protein folds in on itself, which directly affects its function. Even one change in an amino acid, because of a mutation for example, can change the entire shape and function of a protein. So let's walk through the whole journey again. DNA holds the original instructions. Transcription creates a matching strand of mRNA in the nucleus. mRNA is processed and leaves the nucleus. Translation begins at the ribosome, using tRNA and rRNA to assemble amino acids. A protein is made and folded to do its specific job within the cell. This process, which we call gene expression, is how your DNA becomes who you are. RNA is not just important in biology, it's revolutionizing medicine. For example, vaccines, like the ones that fight viruses, use tiny pieces of RNA to teach your cells how to recognize a particular virus without ever using the virus itself. CRISPR, or CRISPR, uses RNA to guide gene editing tools to exactly the right spot in the DNA, allowing scientists to fix certain broken genes. RNA interference can silence faulty genes that cause disease by blocking their messages. These technologies show just how powerful RNA really is. At this point, you should be able to generate a full and appropriate response to today's essential question. What are the functions of mRNA, tRNA, and rRNA, and how do they work together to make proteins?